So this is a basic diagram of the lac operon. Lac is short for lactose. It's a pentose sugar. And basically just imagine this isn't a prokaryotic cell. This is a form of gene expression. A lac operon is a unit of gene expression within your DNA. It's very easy to analyze it within a prokaryote because you have plasmids. So you have circular DNA, only one coil, and not many coils actually. And so then this is what you'll see in one of them, and it's very easy to analyze rather than in eukaryotic cells. So basically in one operon, specifically this lac operon, you'll have what we call a prog, which is just, it makes up the components of the operon. So you have the promoter, which is here. This is a section of DNA that codes for the regulatory or the repressor protein, and it acts as an enzyme, so it just kind of sticks onto the operon, onto the operator, which is the second component. The operator is basically the on and off switch of the lac operon, so when it comes off, it'll be turned on, and when it comes on, it'll be turned off. So basically this form of gene regulation is, this operon is called an inducible operon. So basically, the next component is the genes, which it codes for all the regulatory genes or all the proteins that are needed within the cell. So you would ask, why do we need a lac operon? Well, basically, we don't want to be coding for these genes that later we know codes for beta-galactosidase, which hydrolyzes the reaction of lactose into glucose and galactose. So we don't want to code for that if there's not any lactose, which we can see represented by these pentose shapes, if they're not in the cell. So we don't want to waste energy building those molecules, those very complex molecules with tertiary structures. So once lactose is present in the cell, one lactose molecule will come over to the allosteric site, that's what we know it as, on the repressor protein, and then it'll detach because it'll go through conformational changes. It'll detach, and this is open. It'll allow RNA polymerase to go through the lac operon, and it'll be able to allow mRNA to be synthesized. So mRNA will later code for, it'll go with tRNA and all that process, protein synthesis to make proteins that will later synthesize beta-galactosidase. So it'll hydrolyze the reactions, and it'll break down lactose into molecules that the actual prokaryote can use.